Hi everyone, my name is Rahani and today I'd like to talk about the issue of germline gene therapy in the larger context of genome editing and the ethical controversy surrounding it. I'd like to start off by giving a little bit of a background and description of specifically what germline gene therapy is in the larger context of gene therapy and genome editing as a whole. Specifically, genome editing involves the alteration, deletion, substitution of genes in order to treat the expression of a diseased gene. Specifically, in the case of the therapeutic uses of genome editing involve gene therapy, and gene therapy involves treating a disease. Gene therapy normally occurs in somatic cells, but germline gene therapy involves occurring in germ cells, so that the changes in the gene can be transferred to the offspring. And the essential process of gene therapy involves first of all, extracting a diseased gene from an individual, inserting a healthy gene through a viral vector or through essentially recombinant DNA into the cell of the diseased gene and reinserting that into the individual so that the offspring of that individual and their descendants no longer have the expression of that diseased gene. And so the mutations are no longer present in the offspring. It must be FDA approved to be carried out, and the NIH discusses the current progress, and the clinical trials are currently in progress, but a number of ethical issues remain, which dictates and largely affects whether these clinical trials can continue to take place and whether germline gene therapy can be considered an ethical practice. Again, germline gene therapy involves the extraction of cells, insertion of a healthy gene, and placement within the individual so that the offspring of the individual have a healthy copy of the genes and no longer express the disease trait. Let's take a second to talk a little about genome editing as a whole, specifically in the context of a CRISPR-Cas9 technology. You may ask, what is the relevance of this to germline gene therapy? Talking about the ethical implications of this technology might actually give a very good background into the ethical issue of gene therapy. I'd like to take a second to stop the share at this moment, and actually I'd like to begin the share, and I'd like to take a look at a website, and this is a website that I created myself with, of course, the help of the website designer, which has to do with the CRISPR-Cas9 technology and various ethical issues involved. Specifically, some of the ethical issues have to do with similar ones that also align to germline gene therapy. One of the main ethical issues that is common between the two is the fact of mutations, and the fact that mutations can be passed on to offspring unintendedly, within an unintended fashion. And that is precisely the reason that there's ethical controversy. Specifically, the CRISPR-Cas9 technology talks about genetic enhancement and has to do with this whole concept of designer babies, and that is one of the potential ethical controversies of this issue. And some of the issues surrounding that also happen to be unregulated mutations, as I previously mentioned. The potential of the offspring to have mutations, non-disease related, radically increases when gene therapy takes place. Germline gene therapy takes place, not in the case of somatic cell-associated gene therapy. And so in order to prevent these off-target gene regulations and the gene alterations, the only way would be to not perform germline gene therapy, but I'll get more into the ethical issue in just a moment. Another one of the aspects of the CRISPR-Cas9 technology, kind of contrasting it with germline gene therapy, is that it does involve non-therapeutic modifications. So previously mentioned, designer babies, genetic enhancements, which are non-therapeutic. However, germline gene therapy focuses mainly on the therapeutic modifications, disease treating. I'd like to go back and share the screen once again and talk a little bit about the specific arguments associated with this ethical debate of germline gene therapy. I'd like to take a look and first mention that some of the controversies, in addition to the altered deleterious genes or essentially harmful mutations that can be passed on to offspring, non-disease associated mutations, have to do with genetic enhancement. And similar to how genetic enhancement can create unfair social stigma against individuals expressing a genetic defect, in this case a disease-causing mutation in their phenotype, in the case of germline gene therapy, individuals expressing a certain disease phenotype can also unfortunately have to deal with a very unfair and unjust social stigma. In addition, the economic divide that exists between individuals who can afford gene therapy, between individuals who cannot, also is a major reason that germline gene therapy is part of a major ethical controversy at this time, and clinical trials are currently on a halt and being reconsidered. Lastly, germline gene therapy does not necessarily ensure patient autonomy because of the potential consequences on the offspring, as well as the fact that the entire process is not exactly fully and been clinical trial and tested. So in the full and complete sense, patient autonomy cannot be guaranteed to necessarily be met. I'd like to go a little bit of a depth in the argument itself, and this is the first argument, which is against germline gene therapy regarding an unintended mutation that could be caused. The argument is that germline gene therapy is unethical because germ cells, when passed on to an individual's descendants, open up the possibility of offspring developing harmful mutations. Premise 1, germline gene therapy means that the genes passed down to the descendants can develop mutations. Premise 2, genetic mutations are hazardous for the health of the organisms. Premise 3, if any harm can be prevented from occurring, necessary steps should be taken to prevent it. And finally, the conclusion is because there is a way preventing this harm from occurring, the necessary steps taken to do so, in this case not killing the practice of germline gene therapy, should be the opted for choice. And the Kantian perspective aligns with this, as well as the next argument. And the next argument has to do once again with patient autonomy. Humans whose genes are involved in gene therapy should be able to exercise their rights of autonomy, or the right to be in control of what happens to their body, and if a certain practice does not ensure this, it should not be permitted. Premise 1, the right of autonomy ensures that every individual should be permitted the right to dictate essentially be in control of what happens to their body. Premise 2, germline gene therapy does not ensure full control to the individual carrying the disease gene, in this case the parent of the offspring who is being prevented from expressing the genetic disease phenotype. And finally, the conclusion of this is that germline gene therapy should not be permitted because it does not necessarily ensure the rights of patient that are involved in patient autonomy in a full and complete sense. And it should only be allowed if those rights are met. Finally, I'd like to just cite some sources, the NIH, multiple genome editing databases, and specifically the germline gene therapy Science magazine and NIH sites. Finally, I'd like to thank the website sponsor to help me develop the video and the website design. And thank you, Professor, and thank you, fellow classmates. Thank you.